All right, hi everybody. We're going to continue with macromolecules. We're going to talk about lipids. In the previous one, we talked about intro to macromolecules and dehydration synthesis and hydrolysis and carbohydrates. This one should be a little bit shorter. So today we're talking about lipids. Lipids, the term lipid is synonymous with fat. So the lipids are the fats, the oils, and the waxes, and you're going to learn about hormones and steroids are also types of fats. They are also, like the carbohydrates, made of carbon, hydrogen, but this time much fewer oxygens. Last time we were C6H12O6. This time we're going to have lots and lots of carbons and hydrogens and almost no oxygens, and that's how you're going to start recognizing them. The lipids are long-term energy storage. So last time we mentioned that some of the, especially the polysaccharides, can be used for short-term energy storage. If we really need to store energy, in other words, we're eating way more food than we're burning and our body is wanting to store that energy, that's in lipids. Each lipid molecule has 2 times the energy per gram than the carbs do. And there are no polymers in this case. There's no monomers and polymers. So all the others have like building blocks. Lipids don't have polymers. They don't have the building blocks. And there are 3 major categories. Get my picture out of the way here. There are the triglycerides, we'll talk about first, the phospholipids, which you find in cell membranes, and then the steroids, which include hormones. So let's go ahead and talk about them. First of all, we got to talk about the saturated and unsaturated fatty acids. So there's several things that I want you to notice. First, notice the functional group that's in red on the far left on both of these. So that's the carboxyl acid group or the carboxyl group, and it's where the name acid comes from, where the fatty acid comes from. So it's a way of identifying that this is going to be a fatty acid. Fatty acids are really, really, really long chains of carbon, 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 carbon with hydrogens all the way around it. Saturated fatty acids have no double bonds. My picture is in the way. Have no double bonds in the carbon-hydrogen chain between the carbons and hydrogens. So if you look over here, carbon is single bond, single bond, single, 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 single. So saturated fats have single bonds between all of the carbons. Now, hopefully some of you are like, well, KJ, I see a double bond. Well, that double bond in the carboxyl group, that's not between carbons, that's between a carbon and an oxygen. And that one is always there, and it doesn't matter whether you have a saturated or an unsaturated fat. So the next thing I want you to know about saturated fats is that they are not healthy for you, and they are solid at room temperature. They usually come from animal fats, like butter, for example. And so the way that I want students to remember this is saturated fats have all single bonds, and they are solid at room temperature. So all the S's put those together. So you can see all the single bonds in the saturated fat. It's also possible to draw these as just a bunch of zigzags. So they often will look like the zigzags. Remember that skeleton shorthand way of drawing it, where there's a carbon at each vertex and then there's implied hydrogens that are there? That's what usually the way scientists will draw out fatty acids. The unsaturated fatty acids, as I'm sure you can guess, have at least one double bond. So there's the double bond. It's circled in red. And the double bond actually causes a kink in the fatty acid itself, so it causes it to bend. They tend to be healthier, although the trans fats are the worst of all, and they've been by and large removed from the American diet. So most products, if you look, you'll see that they don't have trans fats anymore like they used to. The unsaturated fats tend to be liquids at room temperature, and they tend to come from plants. Substituting unsaturated for saturated often reduces cholesterol levels in folks that are especially prone to high cholesterol. So a lot of that is actually genetic also. But when you've got those bad genes, then trying to control it with diet by using the unsaturated fats can be helpful. All right, moving on. So now all of those fats can be glued together to a molecule that's called glycerol. So I want you to look at this. This is the glycerol molecule right here. And all of these long chains that were added to it, how do you add a whole bunch of molecules together to build something bigger, everybody? That's dehydration synthesis. So this is one fatty acid right here. It's a saturated fatty acid because it's all single bonds. And it was added to the glycerol molecule, and a water molecule was pulled off. That's dehydration synthesis. Then another one was added, and a water molecule was pulled off. And then a third one was added, and a water molecule was pulled off. So dehydration synthesis. This was the original glycerol molecule, and then we added 3 saturated fats. So if you eat more calories than you burn, the extra energy may be stored as triglycerides in your fat cells. Low triglyceride levels are correlated with healthy hearts. Now you know, triglyceride. All right, next are the phospholipids. As you can imagine, phospholipids have phosphate in them. So see if you can track down the phosphate. There it is, or the phosphorus, I should say, and then the whole functional group with the 4 oxygens, that's called a phosphate. So there it is. And what it has underneath it are 2 fats. So we just came from the triglyceride that had 3 fats. Phospholipids just have 2 fats hanging from them. Phospholipids are attached to glycerol also, and they are the main components in cell membranes, wickedly important for all living things on this planet. So cell membranes are made of 2 layers of phospholipids, and you can see this image here, and I wish you could look at it really close, so maybe scooch in and look at it really close, but those are the phospholipids. They're often drawn, phospholipids, like this is a really complicated molecule. They're often drawn with a circle, and that's the hydrophilic head. It's hydrophilic primarily because of that phosphate group, but also because of the glycerol molecule. That's hydrophilic. And then the tails are hydrophobic. That means they don't like to be near water. So what happens is if I were to take, now you can't really do this, but let's just theoretically, if I were to take a whole bunch of phospholipids and I were to throw them into a bucket of water, the heads are attracted to water, so they're going to immediately turn and face toward water. The tails are afraid, hydrophobic. They're not really afraid at all. They're just repelled by water, and so they turn in. So the heads go toward the water and the tails head toward each other. So take a look at this image of the cell that's right here. There's water on the inside of the cell, so all the heads of the inside layer are pointed toward the water. And then there's water on the outside of the cell. So the second layer, the heads are also pointed out, and all the tails are pointed on the inside, and that's where there is no water. So that just happens automatically, and that's just their reactions to being polar and nonpolar. So the head is hydrophilic and the tails are hydrophobic. All right, and then the last category of lipids, yes, we're on the last page, everybody, are called the steroids. The steroids, one of the class of hormones is a steroid, and then cholesterol is also a steroid. So I've got some images here of what the steroids look like. I want you to pay really close attention. This particular structure is what they call a fused ring structure. So if you remember back when we were looking at the carbohydrates, one of the ways that you were going to recognize the carbohydrates is their rings. The monosaccharides have 1 ring, the disaccharides have 2 rings, and the polysaccharides have a whole bunch of rings. So these guys have a whole bunch of rings. KJ, what are you talking about? Please go back and look at polysaccharides. All the polysaccharides, they have a ring. I'm trying to do this in front of you. They have a ring, and then they have a little like V with an O in it, an oxygen in it, and that's called the glycosidic linkage. They are not actually fused together. There's this little link in between them. That's what a carbohydrate looks like. But if you look at the steroids, they're a type of lipid, they're actually fused together. Like there's no linkage in between them. They're fused together. And that's how you're going to recognize the steroids. So be careful to differentiate them from carbs. The presence of functional groups, this is interesting. If you look at how similar they all are, look at how incredibly similar those molecules are. It's the presence of the functional groups, the little areas that are different, that determines their function, anywhere from being cholesterol to being vitamin D to being progesterone, I think is down here, and estradiol, which is another female hormone. Those all vary depending on the functional groups that are attached. I think that's it, everybody, if I'm not mistaken. I'm just going to double check. Yeah, we're up to proteins. Have a good night.